Now let's take a look at the data report for the physical transport lab. The Brownian movement, experiment A, you didn't get to do. If you would have done it, it would have looked like putting a drop of milk that was boiled and a drop of India ink to mix them on a slide and take a look under the microscope. And it really takes a while before you see these little suspension particles in the milk that they increase their bounciness, if you will, when you're heating up the slide. The first question here says, how could you demonstrate a Brownian movement is not evidence of life? In other words, somebody could say, well, what you see dancing around there in the milk is not little milk particles or oil droplets in the milk, but it's maybe bacteria. And that's for the reason to eliminate that possibility, we boil the milk. So we can say, well, whatever movement you see there is actually Brownian movement, or this molecular movement. Of course, you can't see molecules, but you can see the little milk suspension droplets. But anyway, so what effect did heat have on the movement? So you were supposed to then heat up the slide, and all you see is really that bounciness in these little suspension particles that it increases. So an increase in the Brownian movement, and Brownian movement is really a measure of heat and energy in the system. Moving on to experiment B. Diffusion of a solid into a semi-solid agar. Hopefully you got a chance to look at that video. In that video, they had 3 dyes. They were using an additional one. I think it was saffron or something in red. We usually only use the methylene blue and the potassium permanganate, which you have right here. And I'm going to give you some fake data, and we'll create a fake graph of what that might look like. So here's your fake data, and we're starting out usually with a well that's 8 millimeters in diameter, and so that's what I put right here, these 8 millimeters right here. And then you will see that the methylene blue definitely diffuses a lot slower into the agar, and the potassium permanganate diffuses a lot faster into the agar. And so you have a faster increase in diameter here, clearly with the potassium permanganate, and a slower increase in diameter in the methylene blue. And here I've just put the data here for the first hour, so 15 minus the 8 original millimeters. That would put us at 7 millimeters per hour increase in diameter. And here for the potassium permanganate, for the same time increment, you have 11 millimeters per hour. So which dye diffuse more rapidly? Clearly the potassium permanganate, KMnO4. And what's the relationship between molecular weight and the rate of molecular movement? You can see the molecular weight here for potassium permanganate is 158, and for methylene blue is 342. So the relationship is that the higher the molecular weight, the slower the rate of molecular movement is, or diffusion is, or conversely, the smaller the molecular weight, and the higher the rate of molecular movement would be, or diffusion in this case. What term is used to describe the movement of molecules from an area of greater to an area of lesser concentration? And that term is diffusion. Okay. And why do the dye molecules move? Well, they have energy, so there are collisions, so there's this Brownian movement, and so therefore we have a concentration gradient, and we have heat in the system, so that's why they're diffusing. So why do they move due to Brownian movement? Because there's a concentration gradient. So here, concentration gradient / Brownian movement. Now here we're supposed to construct a graph, and I'm gonna do this. Put you on pause real quick. So here's what the data that I gave you in this table right here, table 3.2. This is what they would look like in this graph. The timeline, of course, the x-axis, and then we're starting here at 8 millimeters. It's our 0 times 0 point, and you can see that the methylene blue clearly diffuses slower than the potassium permanganate, which is in red up here. And you can also see that both dyes will level off, so the rate will decrease after a certain amount of time, just because the diffusion distance is getting bigger, and the concentration gradient sort of slows down too as you reach a certain distance. But anyway, so that's a pretty typical kind of diffusion graph. Moving on to experiment C, filtration. I don't know if you got a chance to do this, but if you filter coffee or tea, you will see that the soluble parts you will end up in your beaker or in your cup, and the insoluble grinds or the tea leaves, they will stay back in your filter paper. In the lab, we would have had a mixture of ferric chloride, which completely dissolves as a salt, and it's yellow and it shows up on the bottom of the beaker. And then we have the charcoal clearly getting stuck in the filter paper because you can see the dark little granules from the charcoal. And then the starch also gets stuck in the filter paper, but you can't really see it because it's white starch particles on white filter paper. And so we have to use an indicator solution. We use a drop of iodine to drop that onto the filter paper to demonstrate the presence of the starch particles, because the filter paper will turn dark blue when you're putting a drop of iodine onto that spot where the starch is. Anyway, so let's answer the questions. Question 8, what happens to the filtration rate as time passes? Definitely slows down, because filtration is a process that it is passive, but it occurs as a result of some physical force. And in the case of filtration, it is the hydrostatic pressure. In other words, you have that column of water that's pressing down on the filter paper. And as that column or this weight, the mass of the water decreases, the filtration slows down. Also, starch particles, for example, they tend to clog up the pores of the filter paper, and that also can slow things down considerably. If you filtered coffee, then you would also notice that in the beginning, the coffee, it runs a lot faster than as the water column in the filter paper gets smaller, then it slows down. Which substances or substance pass through the filter paper? So in a real lab experiment, only the water and the ferric chloride would have passed through. So it's the FeCl2. In the case of if you did coffee or tea, then like the caffeine is water soluble, and other substances from the tea leaf are soluble. They end up on the bottom, and then the tea leaves themselves or the coffee grinds, they wouldn't make it. They were stuck in the filter paper. So on 10, some molecules pass and others don't. The reason for that is that only what's small enough to pass through the filter paper pores will end up going through and end up on the bottom in the beaker or in your cup. So the reason is the molecular size or the particle size. So on 10, our answer is the molecular size or particle size and the filter paper pore size. On 11, based on your results, approximate the relative size of the filter paper's pores. You have some information given, but these are not microfilters that we have in the lab, and so it's not really awfully important to estimate the size here very accurately. So about 100 to 10 to 100 micrometers maybe, based on the fact that starch particles don't pass through the filter paper pores, and starch particles, they are maybe about 10 micrometers, somewhere around that, 10 to 100 micrometers. On 12, where in the human body does filtration occur? So the obvious place would be the kidneys. There are other places too as well, the liver, the spleen, all your lymph nodes, the cerebrospinal fluid is all the time filtered. So there's various places where filtration occurs. Experiment D, the effect of osmotic pressure on red blood cells. So you were supposed to take a look at that video, and they're doing exactly the experiment that you would have done in the lab, and it's kind of cool that they're showing it under the microscope, because it's really very nicely done there, and you can follow the bursting of these red blood cells, which is pretty cool. So the appearance of red blood cells under the microscope we usually use 400x total magnification. Sketch the appearance of the red blood cells below and label accordingly. So let me do this real quick. So when you're placing red blood cells in hypotonic solution, it means 0% salt, which is pure water, then at first they will swell up and then they burst. And that's why I didn't draw anything there, because they're gone. They completely disappear. You don't see anything under the microscope anymore. In isotonic solution, it's 0.9% salt. You will see nice round donut-shaped red blood cells, and they look pretty happy. And in hypertonic solution, a 5% solution, that's what we have in the lab usually, they end up shrinking up, and that's because they lost a lot of water. Okay, so now let's explain the terms below. Hypotonic, that means that a solution that I have in front of me has fewer dissolved particles than the one I'm comparing it to. So if I compare water, pure water, to 5% saline, then the water is hypotonic because there's fewer dissolved particles compared to the 5% salt solution. Isotonic. Tonicity means strength, iso means same. So there are two solutions, and I'm comparing them, and they have the same amount of dissolved particles. So that's when you are placing your red blood cells into normal saline, that's 0.9% saline, then they are isotonic to one another, and there's no net movement of water in either direction. Hypertonic means hyper means above, tonicity strength. So the solution I have in front of me has more dissolved particles than the one I'm comparing to. So I call it hypertonic. Keep in mind that the terms are relative, and you are comparing two solutions, and it doesn't matter what you're starting out with. If you're comparing 5% saline to another 5% saline, then they're still isotonic to one another. So it's relative to one another, okay? Hemolysis and crenation. Describe the result of the 3 small vials. Explain their observation. So if you were to put a drop of like sheep blood, that's what we have in the lab, if you put that into distilled water, then the red blood cells disappear very quickly, and you have a clear test tube in front of you. And that's the same as what happened under the microscope. These cells burst and then they're gone. You can't see them anymore. Crenation happens when these cells lose water, so they look like this. This is crenation right here. That's what happened to that poor little cell. Lost a lot of water. And that happens when you're placing the red blood cells in 5% salt solution. They lose a lot of water, and that's crenation. Okay, but visibly you can't really see anything in a test tube because the cells are still there, and they create that turbidity, and you can't really see the cell size too well. Under the microscope you can see it. Okay, number 16, what's the name for the movement of water when it crosses a semi-permeable membrane? And that's called osmosis. Osmosis. And 17 is 10% sodium chloride, isotonic, hyper or hypotonic to the fluid in red blood cells. So keep in mind RBCs are 0.9% saline. So that means the 10% is clearly hypertonic. Describe what will happen to red blood cells when they are placed in 10% salt. Well, they lose a lot of water, so the water will leave the cell, and this condition is called crenation. Number 18 is 0.009% sodium chloride isotonic, hyper or hypotonic to the fluid in red blood cells. Well, here in this case, we're still, the RBCs are still 0.9%, so we are now hypotonic. Describe what will happen to the red cells when they are placed in 0.009% salt. Well, they will gain water. Water will rush into the cell, and that condition is called hemolysis. Hemolysis, the red blood cells will burst. So just to be sure that we're on the same page, I drew two beakers here. Let's go on 18 here first. So the situation is that we have 0.9% salt inside of the cell. That means we have 99% water. And osmosis, remember, that's the movement of water down a concentration gradient. So if we have 99.1% water, compared to 100% water on the outside, 0% salt. So we are placing these red blood cells in distilled water, or what is it here, 0.009% anyway. So at any rate, if the water concentration is greater on the outside, then the water will go inside of the cell, and animal cells don't have cell wall, so they will burst. Up here, by contrast, if we go back to 17, we have 0.9% salt still in the cell, and on the outside we have now 10% salt, and so now we have 90% water, and 90% water is definitely less water than 99.1% water. And so in this case, the water will go also from high concentration to low concentration. In this case, the cell will lose water and it will shrivel up, and we call that crenation.